Okay, let's dive into this. You know, viruses are just masters of survival. They're constantly changing, adapting. But what's so fascinating is that their greatest strength, this ability to mutate like crazy, well, it hides a massive, potentially fatal flaw. It's a story about living right on the absolute edge of disaster.
So, let's start with this question, really let it sink in. What if the very thing that makes viruses so tough to beat, their ability to change at lightning speed, was actually their biggest weakness? Now, this isn't just some random thought experiment. This is a real, central vulnerability that scientists are learning how to exploit.
In this slide just lays out the massive difference in strategy perfectly. You've got DNA-based life over on the right. That's us. Our replication is super careful, really precise, because we have all these built-in proofreading tools. But the RNA viruses on the left? Oh, they are playing a completely different game. Their whole replication system is fast, it's sloppy, and it has absolutely no spell checker. And this error-prone process is what creates this incredible amount of genetic diversity, which lets them jump to new hosts and dodge our immune systems.
But, there's a limit to how messy you can be. Way back in 1971, a chemist named Manfred Eigen came up with this idea, the error threshold. Think of it like a genetic speed limit. A virus needs some mutations to survive, that's a given. But if it mutates too much, too fast, it basically starts to lose its own identity. All that essential genetic information that makes it a working virus just gets scrambled into complete nonsense.
And when a viral population crosses that line, it doesn't just kind of fade away. Nope, it triggers what we call an error catastrophe. It is a complete and total genetic meltdown. The population gets so filled with bad mutations that it just can't reproduce anymore, and the entire lineage just collapses. So that's the theory, right? It's clean, it's elegant. But as scientists started plugging in real-world data, a much more complicated picture started to emerge. Was it really a sudden, sharp cliff that a virus just falls off of? Or was it something a little fuzzier?
And these images show us exactly what that fuzziness looks like. Over on the left, that's the simple theory. As the mutation rate increases along the bottom, the main virus population, that black line, just suddenly vanishes. It literally falls off a cliff. But the graph on the right? That's what happens when you factor in all the randomness of the real world. That sharp cliff just disappears, and it's replaced by a much more gradual slope. We call this the crossover region.
So, the takeaway here is really important. That blurry slope, that crossover region, it kind of gives the virus a safety net. It makes it more resilient. It's not teetering on a knife's edge anymore, which for us makes it a whole lot harder to just give it a little nudge and push it into that error catastrophe.
So what's actually causing this blurring effect? Well, scientists ran the models to figure it out. And while random differences in how fit each virus is does play a part, the real driver is something else. Just look at that last bar. It's the randomness in the mutation rate itself that makes the biggest difference. The speed of mutation isn't some constant number. It fluctuates, and it's those fluctuations that really stretch that cliff out into a long, blurry slope. Now, this error threshold doesn't just put a limit on the mutation rate, it also acts as a hard cap on how complex a virus can be. As Edward Holmes points out here, because you can't have too many mistakes, you also can't have a very long genetic code to make mistakes in. It creates this kind of natural size limit for almost all RNA viruses.
And that limit is right around 15,000 base pairs, or 15 kilobases. For the vast majority of RNA viruses out there, this is the ceiling. They just can't keep their genome stable if they get any bigger than this. It's like a fundamental rule of their biology.
And then there are coronaviruses. Get this, at around 30 kilobases, their genomes are literally twice the size of that supposed limit. They are the absolute giants of the RNA virus world, and they're a huge, glaring exception to a rule that holds true for almost everybody else.
So this immediately begs the question, right? How on earth are they pulling this off? How do you maintain a genome that huge when you're using the same error-prone copy machine as everyone else? They have to have some kind of secret.
And here it is. It turns out coronaviruses have something that's unique among RNA viruses, an enzyme called exoribonuclease, or XON for short. This is their secret weapon. It literally functions as a proofreader, a biological spell checker that follows along behind the sloppy replication enzyme and cleans up all its mistakes. This is what lets them have a much larger, more complex genome without just collapsing into error catastrophe.
And this diagram just shows that internal dynamic so perfectly. It's a constant tug of war.
You've got the main replication engine, the RDRP, just blazing ahead, making copies and making errors, but right behind it, you've got the XON proofreader, fixing those mistakes as they happen. It's this delicate balance between speed and accuracy that lets coronaviruses have the best of both worlds, really fast replication and a large, stable genome.
So, what can we do with all this information? Well, understanding this delicate dance between mutation and collapse isn't just for academic papers. It actually opens up a really powerful new strategy for fighting these viruses by, you guessed it, turning their own biology against them.
This strategy is called lethal mutagenesis, and it's basically fighting fire with fire. We know these viruses love to live dangerously close to their error threshold, so the whole idea is to introduce a drug that intentionally ramps up their mutation rate, pushing them right over that limit and forcing them into a full-blown error catastrophe. Instead of trying to block the virus, you make it mutate itself to death. And this brings us right back to where we started. Remember that blurry slope? This is why understanding it is so incredibly critical. To make this lethal mutationist strategy work, it's not enough to just nudge the virus over the start of the threshold. No, you have to give it a high enough dose of the drug to push it all the way past the upper limit of that crossover region. That's how you guarantee a total population collapse.
So it's pretty clear we've found a fundamental weakness, a real Achilles heel in the armor of these RNA viruses. But this is a constant evolutionary arms race. They adapt, and we have to adapt our strategies. Which really leaves us with a final thought. What new vulnerabilities and what new viral tricks are waiting for us just over the horizon?